Yes, sir. Please start.
All right. Very good morning, everybody. Welcome to the first live session for this MOOC. Welcome to all the students who are joining us today for Proteomics course, Proteogenomics course, and Proteins and Gel-based Proteomics course. I hope you have gone through all the videos of the initial month and started knowing the basic concepts of these technologies. The field of proteomics is growing very rapidly. However, the basic concepts and basic technology, they're always going to be applied and very useful, especially if we talk about gel-based proteomics, which is the theme for today's session is. Gels have been used from decades, at least we know that 2D gel electrophoresis was discovered almost 50 years back in 1975 by scientists Close and Farrell. And from that time onwards till now, if we think about, we are actually looking at different ways of simplifying the complex proteome, and gels are always being used, despite that there has been advent of many technologies, especially mass spectrometry, have played very important role. But nevertheless, different type of gels are still one of the very fundamental and important first activity for doing the proteomics experiments. In today's live session, we are going to give you the live demonstrations from the lab for various type of gel-based proteomics workflow, starting from the concept of the sample prep, taking forward to do the quantification, moving on to SDS-based gel, 2D gel, and different applications from that. So that will be the today's focus. But before I start, I'm sure you already know that I'm the instructor of your course, and I'm a professor at IIT Bombay here in Department of Bioscience and Bioengineering. My lab does a lot of work on clinical proteomics when we apply the proteomic technologies to understand different type of diseases. We try to identify biomarkers for detection of diseases early, or to look at how disease progresses, the prognosis of the disease. In that light, we utilize different type of proteomic technology platforms, from the gel-based to the mass spec to microarrays, etc. We work on many problems, especially in several type of cancer we work on with Tata Memorial Hospital and many other collaborating hospitals, especially on the brain tumors, but also on the prevalent cancers of India like breast, cervical, oral cancer. And then additionally, lab has also done a lot of work on the infectious disease, especially COVID time. We have investigated COVID-19 problem and tried to look at the prognostic biomarker which can indicate a patient moving from mild to severe form. And again, like despite the diseases are all different type of complication, but the technology platform remains same, and the same technologies could be applied to understand any kind of clinical or biological question. Therefore, I think these courses what you are taking is going to pay you off at some point for sure in your academic and research journey, whether you will take proteomics as the core or not. But even for any kind of biological or clinical application, you will be required to use these approaches. So with this, first let me have introduction of our team, like the TAs who are going to take today's live session with me, and who are already part of the course, who are interacting with you on the forum and for different interaction activities. So I'll hand over to Shashwati, and she and the TAs are going to first introduce themselves so that you can know that with whom you are working and what their backgrounds are. All right, Shashwati.
Yes. Good morning, everyone. So I'll just share my slide. Yeah. Good morning, everyone. Myself Shashwati. I am in TAs in proteins and gel-based proteomics. So I have joined IIT Bombay under Sanjeeva Srivastava in 2019. My research mainly focuses on multi-omics investigation of cellular adaptation, which drives cisplatin Nelson in triple negative. So I mostly work on the TNBC. It's a very heterogeneous and very aggressive breast cancer subtype. So I look into the different aspect of the cisplatin resistance because it's mostly given to the patient, and it increases the resistance after some time. So that's why my goal was to understand what is the metabolic adaptation occurring in the resistant patients. So that's why I'm working on the multi-omics aspect of it. So then I'll hand over to the Ankit, our next TA.
Yeah, hello everyone. Good morning. So I am a TA for the course of Introduction to Proteogenomics, and I am a final year PhD student here at IIT Bombay. So my work mostly is on the multiomics investigation to understand and elucidate different pathophysiology of meningioma and to find different novel candidate markers. So feel free to ask if you have any queries in the discussion forum, as well as we have a Telegram group, so you can join and discuss. Yeah, thank you.
Good morning, all. My name is Amit, and I am the course TA of Introduction to Proteomics NPTEL course. And I am in my third year of my PhD, and my project is based on comprehensive multiomics investigation of pituitary and neuroendocrine tumors, where my goal is to find diagnostic and prognostic markers of this neuroendocrine tumors. So there are a lot of subtypes of these tumors, like acromegaly, Cushing, and NPA, and my objective is to find subtype-specific metabolite, lipidomic, or proteomic-based markers. And there, for the introduction to proteomics course, there is another Telegram group. And if you have any doubt, you can always ask in the discussion forum. And also, you can join the Telegram group for the discussion. Thank you.
All right. So now you know that your TAs are quite competent research scholars, and when they are talking to you, they're talking with a lot of experience. So please do utilize their expertise of research for over the years, and interact with them whenever there's opportunity for the your forum activities, your Telegram groups, or even the live sessions. What we thought, since today the first time which we are meeting, we will show you where we work and how a research lab looks like, what kind of technologies we have. So Shashwati, I request you to just give a tour of the lab before we start the some formal session today.
Yeah. Again, good morning, everyone. So I'll show a little bit tour of our lab. So starting with the mass spec instrument. So we have the mass spec instrument in our lab. So first is Q-Exactive. You can see this is coupled with UHPLC. So here you can do multiple applications, like you can perform proteomics or metabolomics as well, and lipidomics, different omics approaches. Here, for now, we are using for the metabolomics run, which is a small molecule to look into in the different patient sample, or you can use for different serum sample, tissue sample, anything you can use. So here you can see this is a Q-Exactive mass spectrometer, and this is the UHPLC. So here we have our auto sampler where we inject samples. So you can see there are different vials are available here. So you need not to be there in lab. You can just pick your sample and we take the sample from the auto sampler. Then this is the UHPLC, and one run is ongoing. You can have a look. So how the chromatogram for the run looks like. So this is the metabolomics run. Yeah, so here from the auto sampler, the sample goes from here, and here we have the connection where it indirectly goes to the mass spec. So here you can see the ionization is occurring and the mass spec is analyzing our sample. So you can see in the HPLC, we have the column here, which resolves our sample, and further it goes into the mass spec. So this is one kind of mass spectrometer. Then we have another mass spec, which is thermofusion orbitrap fusion. It is a tribrid mass spec. So here we have connected the nano LC coupled with this mass spectrometer. So here in the nano LC, it will flow in the nano gradient. So here you can see, here also we have the auto sampler. So I'll just try to show you where we keep our samples. Basically, we are using this for the proteomics experiment. So you can see this kind of vial we are using for our auto sampler. So here we keep our samples, and you can just keep the Q and it will take the sample. So from here in the nano LC, the sample comes from here to here. So this is the nano column here. So from this column directly it is going inside the mass spec, and you can see here so here you can see the sample is getting ionized and it is in the mass spectrometry, it is getting analyzed. So this is the hybrid mass spectrometer. Usually we use this for our global experiments. Then moving forward, we have another mass spec. It is called triple quadrupole. So basically here it helps with our targeted analysis. So in the triple quadrupole, we have a Vanquish autosam UHPLC. So you can show this is the auto sampler where we have our samples. So you can keep your samples here for the analysis. So it is the HPLC. Okay. Yeah. So again, the sample takes from here and our column is right here. So here we have our column. So from there, the sample goes directly into the mass spec. So this is the system here to the triple quadrupole. So the application of this is mainly we use for the targeted. Now from the global, you have your target list of proteins or metabolites or lipids. You can do the targeted assay here. So after that, we have a different instrument, which is a BLI. So here you can see, so it is mainly used for the ligand and drug kind of binding experiment that you can perform here. So here we have different kind of biosensors which helps in the here. So these are different kind of biosensors which helps in the taking reading in the interferometry, and from that we can calculate the ligand like protein drug interaction. It all happens in the dark condition. That's why we have all the black plates and the black glasses here. So this is a BLI system. Then we can move to our lab. I'll just show you our lab. So in our lab, we have different instruments. So first I can show you, this is the SMC Pro instrument. So you know all about the ELISA. It has a similar application. Like if you have a very low abundant protein, so you can just do the ligand antibody binding assay in this instrument. It will, like in the femtomole also, you can just detect that protein. So it is a very sensitive instrument. So after that, we have the multi-scan reader. So here you have it's a spectrometer. People use usually the cuvette method, but here we have a multi-scan where it can adapt 96 well plate, and you can take the reading of 96 well plate in the visible range. So that's how you can just calculate many sample at a time. So that's the multi-scanner. Then we have a ion torrent system, which is helped in the NGS system. So here you can see this is for the next generation sequencing. After that, I'll show you. Here we have the microarray system. This is the scanner of microarray. So in microarray, we have different slides where like many proteins are printed. So you can see the slides. Yeah. So here in this slide, we have different kinds of antigen which is printed here. So you can just analyze your serum. So it will detect your antibody present in the serum. So I'll just show one slide here, how it will look like after the scanning. So you can see here, there are different spots. So one spot represents one protein, or in the peptide chip, it will represent one peptide. So like that. So if it binds to their antibody, so it will show the fluorescence. So this kind of chip is present here. So after that, we have different kinds of fridge for -80. You can say for the clinical samples, we usually store our samples in the -80. So we have here our -80, where we keep our different tissue samples, serum samples, cell line samples for further use. Now I'll show you our SDS page setup. So here we have set up for the SDS page. This is for the 1D, like it will only resolve on the basis of the molecular weight. So this is the gel setup that will run. So this is the electrode and the cover. Okay. In the upcoming session, like I'll show how to load the gel and how to run it. So for now, this is the setup of the gel. After that, we have the centrifuge here, where we use different kinds of like centrifugation. Also, we have the clusters here, where we do like generally have big data analysis, because in the omics there is big data is required, and for that we need the cluster so that our analysis is ongoing. So that's it about our lab tour. So thank you. In some time, I'll show how we prepare the sample, how we load the gel, and how we run it. So thank you everyone for now.
All right. Thank you, Shashwati. I hope all of you have got a tour of the lab. This is kind of a typical research lab with different type of setups. Doing the clinical work requires a lot more stringency, and especially when you're handling the biological sample, clinical sample, you have to be also considerate that how to ensure the biosafety aspects of your research, right? So those are all part of the training. Let me just give you a little bit overview for today's session, so that it will be easy for you to orient. I'm sure you have been exposed already for some of these concepts. But today in the gel-based proteomics for biological application, today's session, we are going to cover some of the basic technologies like SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis. Followed by, we'll talk about two-dimensional gel electrophoresis. I will not talk today about blue native page, but that is like SDS page, but you are not adding the denaturing conditions. And last, we will cover briefly the 2D dyes, difference in gel electrophoresis, one more sensitive way of doing gel-based proteomics. So this is kind of what we plan for today. Just to refresh you about the concepts, as you are aware, two-dimensional electrophoresis means it requires 2 dimensions to separate the proteome. And our goal here is to look at thousands of protein separation. So since the proteins are, we are talking about a complex mixture, goal is always to see like how many protein we can separate using different properties. So the scientist Claude and Farrell in 1975 thought, can we utilize 2 properties of isoelectric focusing and the PI of the protein and the molecular weight. And for those separation, they utilize IEF for the PI, isoelectric point, and the molecular weight, they use SDS page. Of course, the concept remains same from that time till now, after 50 years, but the overall ways of looking at the setup, from the tube gel and ampoule light to moving to the immobilized pH gradient strips and commercialized setup, all that has changed over the time, but nevertheless, the fundamental concept remains the same. And eventually, after doing the 2D gel, you see some beautiful images like these, which show the different pattern of the protein separated based on their isoelectric point and the molecular weight. And you can stain the gel in different type of staining solutions. So the first dimension separation that requires isoelectric focusing unit, we will show you when I come to the lab. Idea is, when the proteins are moving in the electric field, when they reach to their isoelectric point, pH becomes PI, there is no net charge, and protein stops migrating. And if we are separating protein in the pH range of 3 to 10 or 4 to 7, protein will separate as per their PI values. And then we can separate the first dimension based on their PI, and that is known as the IEF in the field, isoelectric focusing field, for the first dimension separation. Once we have done that separation, then we want to utilize one more property of the protein for separation, which is size. And for that, we add SDS detergent, which makes the protein negatively charged. We boil them, we add the SDS, we add beta-mercaptolithanol to reduce the disulfide bonds. And after all that, when we run on the gel, so this is the sample preparation part, which we'll talk again in the lab setup. So make sure that every protein is denatured, and now you have added the negative charge with SDS. And once you are loading on the gel, then in the electric field, they're moving based on their size. And on the right-hand side, if you orient yourself, the first lane shows the pre-stain markers. So these are the known standard proteins, which we can say this is the 100 kilodalton, 70 kilodalton, 30, 20, 10, 5, like that. So then based on that, we can say, okay, the purified protein, what we can see, looks like something around 50 kilodalton protein. So in this way, you can get estimate of, based on the densitometric analysis, that what is the approximate size of my given protein. So SDS page is also utilize a standalone technique just for the protein separation, checking at the quality of the protein on the gel. And even as I said, even if your goal was to do the mass spectrometry-based proteomics, often you are going to utilize this kind of setup to ensure that the quality of your sample prep is very good. So right now we are talking this in the context of 2D gel, when the second dimension separation is going to happen with the SDS page. But I'm also trying to refer you that even if you don't do 2D gel and you are directly going for mass spec based application, but still many a times when you are preparing the sample, you are going to run on the gel, SDS page gel, to ensure that the quality of the bands and the protein is very good. It is not degraded. You are able to extract the proteome from the high molecular weight to the low molecular weight. And once you are happy with that, then only you proceed forward. Additionally, you are also going to quantify the protein, which we'll be talking in this next session about using the Bradford or BC or other kind of assays. So idea is, if you want to compare in the proteomics-based experiment, your 10 control sample and 10 disease sample. So first you want to ensure that you are starting with the equal amount of protein, because the patients have a lot of heterogeneity. They have their own differences, which we can't avoid, and everybody is unique and having a lot of heterogeneity. But despite all those biological and clinical variability, at least the the experimental variability should not be confounded with that. So to overcome that, we quantify very rigorously to ensure that all our 10 samples of control and 10 of the disease, everything we are going to have the equal amount of protein to start with for our investigation. So let's say you have quantified the protein using Bradford assay, and now when you load on the gel, you are adding, let's say, 5 microgram on each well in the SDS page. And then eventually you can see the pattern that, okay, all these 20 samples which you run in the 2 gels are going to look very uniform. So their band intensities are quite matching. And it is not happening that 2 samples are totally degraded, it's a smear, you don't have anything to see over there. And then you are going to work on a very bad sample. So first you need to do qualitative check and ensure that quantification, what you have done with the Bradford, is also being tested on the SDS page gel. Even if you are not doing 2D gel or not doing the gel-based proteomics, directly want to go for shotgun proteomics, LCMS, which we do mostly in the lab, but still this becomes one of the very important quality control check to look at the qualitative estimation of your protein and ensuring that your quantification reflects well in the quality on the gel. So I hope that clarifies that SDS page alone is very useful. And of course, SDS page is also the second dimension separation for the 2D gel. So let's switch back to the technology of 2D gels, where our goal was to separate from 2 dimensions, when SDS page was the second dimension separation. And we are using the IPG strip or immobilized pH gradient strip to separate our proteins on the first in the first dimension, followed by second dimension. But now when we are ready to load on our SDS page from the first dimension to second dimension, there is a small step here, which is known as equilibration of the IPG strip, because you can't boil this strip. So you are going to do the reduction and alkylation using DTT, dithiothritol, and idostamide. Those steps you have to do on the strip only. So then you can do in solution in the tray. And we first go for equilibration solution 1 and followed by 2 to ensure that all the reduction happens well and it does not oxidize again. So to prevent any alkylation of sulfhydryl group, of cysteine residue, then we add IAA. And once we do these steps, then only we are ready to keep this IPG strip on the SDS page gel for second dimension. So now let's just kind of, view the entire process. We took the IPG strip of 3 to 10 pH, for example. We are doing first dimension separation in the electric field. And after the proteins have, reached to pH equals to PI values, 0 net chart, now we are equilibrating the IPG strip and moving them on the SDS page gel. SDS page, the comb can be different when you are doing the SDS page for one dimension. That time you are going to use the comb. When you are going to use for 2D gel, you're going to use the flat comb, which will just be making it flat space for you to keep the IPG strip overlaying on top of that. So then we do that, and this is the second dimension. But till now, you don't know how the protein look like, so everything is very abstract. After that, you're going to stain your gel, which is very transparent gel, with the different type of dyes are possibility, but the Kumasi brilliant blue, which forms the ionic and non-covalent interaction with the protein, is one of the very commonly and most popularly used stain, both because of it's very robust, it binds the protein very strongly, and followed by the background becomes very light, so you can see the spots very well. It is also very compatible with the mass spectrometry, so protein identification from the gels becomes very easy. And third, of course, the practical way is very cheap as well. So Kumasi brilliant blue we use for the staining the gel, whether SDS page or 2D gel. But often your gels are like, your proteins, what you are going to investigate has many low abundant protein or, a lot of protein which are not visible on the gel. So you can use the sensitive stains as well. And often, since you have put in the clinical sample, for example, a very precious sample, you want to get more information from the same gel. So then people also utilize dual staining method. So here, the same gel is first stained with, let's say, ProQ diamond. This ProQ diamond is selectively stains for the phosphorylation. And once you have done that and scan that gel for the ProQ diamond phosphorylation, then now you can wash it, again fix it, and now you can stain with the Cypro Ruby, which is going to show you all the spots on the gel and much more sensitive than Kumasi. So now from the same gel, one gel only, you are able to, by exciting at different emission spectra, you are looking at 580 and 610, you are looking at just the ProQ diamond based image for the phosphoproteome, and you are looking at the Cypro Ruby for the entire complex proteome global image. The same gel is going to give you both the information for the PTM as well as the global proteome. So this becomes one of the very strong and nice way of doing the applications, especially if you're limited to the samples. But further, people realize that in the 2D gel, we are running the gel separately on 2 different gels. So control sample is run in one gel and treatment sample is run in another gel. And if in our case, we are talking about 10 controls and 10 treatments, it means we have to run 20 gels. Then analyze all of the 20 gels and look for any pattern across each image or the spots. That becomes actually often very challenging because of the reproducibility in running the gel. Because I'm sure if you have made a SDS page gel or run a 2D gel, you know how much dye front mobility movement is not very reproducible and not within your control. It just does not happen 100% perfect. So to overcome that, people thought if we can label our control sample with one fluorescent dye, cyanine 5, for example, and one treatment sample with cyanine 3 dye. So now 2 samples could be done on one gel. So at least we have reduced that complexity. 20 gel became 10 gel. But on top of that, if we can make a pool of all our 20 samples, which represents all our control population and treatment population, all 20 samples we make in one tube, and that we label with the third dye, which is cyanine 2 dye, then this will serve as the internal reference standard sample, the Cy2 tube. And that we are going to run on all the gels. So it means on all the 10 gels, we will have the Cy2 always. But on our first gel, it will be the Cy3, Cy5 pair of C1T1. On the second gel, C2T2. And then again, the Cy2 will always be there. And likewise, till for our C10 and T10, in addition to Cy2 dyes. So therefore, in this manner, we are able to represent our entire population with the cyanine 2 dye in all the gels, which can be utilized for normalization and looking at co-detection of the spots to align the gels very well. And this is going to help us to look for data analysis and reproducible normalization and statistics. Then on each gel, you're only going to have one set of the control and treatment, which you are going to compare. But even to ensure that your experiment is much more robust, you can actually now do the dye swapping. It means if the first set of the gel contains the control has the psi 3, the second one, the control has psi 5, and third one is control is psi 3, and fourth is control is psi 5. Likewise, you are doing dye swapping, so there is no bias for the dyes as well. So this is another technology which is much more reproducible than just the standard 2D. So again, if the goal was to do quantitative proteomics using 2D gels or using the gel-based proteomics, difference in electrophoresis is the way to go forward. And just as you can see the overview slide here, you are preparing your two different sample with two different dyes, psi 3, psi 5. You are making internal standard with the cyanine 2 dyes, mixing all of them in one tube. Now you're proceeding for the IEF, followed by running on the gel. And then the same gel is going to give you three images, one for psi 3 control, for example, psi 5 treatment, and psi 2 internal standard. So this is one of the way of doing the gels much more reproducibly. So I guess what I wanted to just touch upon and refresh to some of you who have already gone through some of this module. Some of you might find it still, it's like, new to you. But we wanted everybody to come on the same page to understand that, gels are very useful for proteomics. Whether your goal was to do the 2D gel-based proteomics or LCMS-based proteomics, gels are the very first step to ensure that what sample you're going to work on, you have the full control of their quality and quantity. And once you do that, then only you can proceed for the next step. Next step could be gels alone for quantification, could be the dyes technology, or you can even think about having your LCMS-based approach for the next quantitative proteomics as well. So let's move on to the lab session now. I don't want to make it another lecture because you're probably bored of watching me on the video. So let's just go to the lab and start the live session of the experiments. So first, we are going to talk about the sample prep and how you are going to take any kind of biological sample, prepare the lysate, and move on from there for doing the quantification and running on the SDS page. So Shashwati, over to you, and let's show in the lab.
Sure. Before showing the live, I'll just share my screen to show what are the steps I'm going to cover. So first I'll show the cell lysate preparation. So the first step would be to lyse our cell or tissue, whatever you have, so that we have our protein from the cells. So there are different methods that we generally use. Like you can homogenize your cell or tissue using Down's homogenizer, or you can use the sonication method, or if the tissue is very hard, so you can either use the mortar pestle kind of method, or you can use the bead beating. So that's the general workflow for cell lysate. So then the next step would be to know how much proteins are there in my sample, right? So for that, we'll do the protein quantification. So there are different methods are available, like Bradford assay, Lorry assay. So mainly the goal is to check the, usually we do the colorimetric test to know the concentration of the sample. So how we do it, we generally draw a standard curve with the known concentration of BSA protein, and from there we plot our unknown samples. So here we have our standard curve, and if you have the absorbance here, so you can just map how much protein we have in our unknown sample. Then followed by that, now we know how much proteins are there in our samples, so we can go for the equal loading in the gel. Otherwise, it will create a biasness if you don't know how much proteins is there in sample 1 or sample 2. Now we know how much proteins are there, so we can load equally in the gel. So there are two types of gels. Already Professor Sanjeeva has explained. 1D and 2D, we're going to show that. So I'll just stop sharing my screen. Now I can show you live. So here in our lab, so first step would be the lysate preparation, which I'll show. Yeah. So here I'm showing the cell lysate preparation. You can use for different kind of fun. So here you can see the cell pellet. So first step would be to clean the cells, like you are growing cells in the media. So previous course we usually do to clean the cells. So I have already cleaned. If you have the tissue samples, then you can just give the PBS wash to remove all the blood particles, because in the mass spec it is going to harm your like it will show only the blood related protein, it will not show the tissue related protein. So for that, first we are going to do the PBS wash. I have already given them PBS wash. So now the next step would be to adding the lysis buffer. So here I have the urea lysis buffer. So I will, according to the cell pellet, you need to judge how much lysis buffer is required. So if the cell pellet is very less, you can give 50 or 100 microliter of lysis buffer. If it's very high pellet, so you can add 200. So now I'm adding 200 of lysis buffer. So you already know what is the composition of urea lysis buffer. So you have to just add the lysis buffer, then gently you have to mix your cells. Then we will go for the sonication method. So, but if you have the tissue sample, then you have to do other kinds of method. So for the sonication, I'll keep the sample in ice. Why? I'll explain in my next step. Here we have the sonicator. So you can see we have different parameters. So for time I have set for 2 minutes 30 seconds. You can adjust according to your tissue or cell complexity. And then we have the amplitude, like in which amplitude we are going to sonicate our sample. So I have set it for 40%. Then we have, you can see pulse. 5 seconds, 5 seconds. That means we'll give pulse for 5 seconds, then pulse off for 5 seconds. Because during the sonication it generates lots of heat. So I'll just start, and you can see that's why I'm giving ice. So make sure your probe just dip into the lysis buffer, otherwise the sonication won't be proper. Then I'll just start. Yeah. So it will do the sonication for 5 seconds, then it will give the rest of 5 seconds, then it will again start. That we are doing because during the sonication it will generate a lot of heat. In that case, the protein of our interest will be degraded. For that reason, you always need to keep your sample inside the ice, and then you have to process it or sonicate it. You have to keep the samples in ice. Okay. So after sonication, I'll just stop it here. So after sonication, you can see the clear lysate, like the cells have degraded. So now I'll add the PIC. So PIC is a protease inhibitor cocktail. So what it will do? So it will inhibit all the protease present in the sample to make sure that our protein is not degraded. Okay. So for that, I'll use the PIC. So how much PIC I'll add? In 1:10 ratio. If I'm adding 200 microliter of urea lysis buffer, then I'll add 20 microliter of PIC. That is 1:10. So you need to add it after the lysis preparation. Okay, now I have added to my sample. Then the next step would be the centrifugation, so that all the debris which is present in the cells, that will settle down and will have the clear lysis in the supernatant. So our interest of protein which are all in our supernatant. So for now I'm doing it in the mini centrifuge, but actually you have to do it in 8000 G for 10 minutes in 4 degrees centigrade. So every time you have to be quick and you have to keep the protein in the cold temperature. Okay, so now you can see my clear lysate. It is all clear. It's not looking any cloudy. And at the bottom, there is a little bit debris of cell which is settled. Now I will take out my lysate. So you can just take out the clear supernatant, and you can discard the rest of the So here I have a new tube where I'm collecting my lysis. So your lysis is ready for the next step. So we can now go for the quantification is these pages. So that's very simple and easy. So you can store the sample for in -80 for further use also. So now we have done the lysis preparation. Then we'll move forward with our quantification technique, like how we know how much protein is there in our lysis. Okay, so for that I have already told you, like we are using different colorimetric tests. So here already I have prepared the standards using the BSA. So we have different concentration of BSA here: 0, 2, 4, 6, 8, 10, 12 and 14. You can take till 14 because after that the curve will not be a straight line, linear line. It will saturate there. That's why you have to prepare within 14 range, so 0 to 14. And here I have unknown. So I have added Bradford reagent to the sample. So this is the Bradford reagent. It looks somewhat like brown color, but you have protein in that, so it will turn to blue color. You can see in the standards it's blue in color. In our unknown also we have the blue in color. So now what we are going to do, will after I have already added the Bradford for you. So you need to add Bradford, then you have to incubate the sample in room temperature for 10 minutes. So after that, you can take the reading at 595 nanometer. So now for the reading, I'll take the reading at a same time. So for that, I'm adding the sample in a 96 well plate, or else you can use the cubate method. But in that method, you have a disadvantage that every time you have to take the cubate, then you have to wash it, and then you have to put your sample. In that way, the incubation for the last standard will be very late, and in that way, your reading will be somewhat biased. So for that, I'm adding the sample in 96 well. Then we can take the reading. So you can just take one reading or triplicate or duplicate. So at least you have to take two readings so that you'll have the exact value. Okay. So you can see the change in color in all the standards. So it's brown at the start where we don't have any protein. Then it's slightly increasing. So for now, I'm adding till 10. Then I'll add my unknown here. Okay. Now we'll show for the scanning. So I'm taking it for our scanning in the multi-scanner. So that I have already shown you. So here we have our multi-scanner. So can you just open it? Yeah. So here we'll place our plate. You can see. Yeah. So now we can just take the reading. So here this multi-scanner is connected to a laptop where we have different plate layout. You can just define blank and your standards. Here I have our unknown sample. So this is the plate layout you have to give. Where is your sample? Where is your standards? After that, you have to set the protocol here. So for the observance, we need the reading at 595 with precision. Then here you have to do the blank subtraction. Then we can just click on start and we save our session. Just a second, you have to save your session, then it will start taking the reading. So you can say now it will show the absorbance here. It will take like 5 to 10 seconds, it's scanning. Then yeah, so now you can see there are different readings are here from where we can plot the standard curve that I have already shown you. So now our plate also has been out from the instrument. Now we can just take out the plate and we'll just keep it. So from here, from the report, you can export your Excel and you can save it. So it will give you a Excel like this, which have all the values like the normal observance and the one with the blank subtraction. From there, you can plot your standard curve and you can know the unknown sample concentration. Okay. So I have already detected my protein concentration, which was around 1 mg per microliter. On that basis, I have prepared my sample for the gel. So this is a SDSP gel. So you can see here we have the comb. I'll just remove the comb here. You have to be very careful while removing the comb, otherwise the well will just not form very closely. You can see I think it's not that visible, but we have the wells here. So I have already prepared my sample for you. Is it visible? Yeah. So in the sample, I have added my protein concentration. I have used 10 mg of protein sample I have taken, and I have added the loading die so that we can track the gel run. So I have made 2 samples here. So now I'll just load sample and show. Yeah. So here I'll show my sample. Yeah, you can take it. You have to locate your well. We have 10 wells here. So here I can add my sample. Yeah, I think it's visible now, right? Yes. I'll show another well loading. So I'll load in another well and show. You have to be very careful so that the sample goes into the right well. It does not leak. Okay. Now we have samples in 2 wells. So for the marking, you can add the protein ladder here, or you can add BSA to know the exact protein molecular weight. So here now I have added already the running buffer. So we'll just connect it. So you can see there are representation red and black. So you have to be very careful while connecting all the electrodes. So the red goes into the red side and black goes into the black. So here we have the gel pad. So I'll just connect you and show. So here you have to put your electrode. Then you have to click on manual. You have to give the voltage where you have to run. Like I have set the 90 volts. Now I can start the running. So if the gel is running, though you can see there is bubble coming out from the gel, so that you can ensure that the gel run is proper, like the gel is running. So you can see there is little bit bubble here. Okay. Now I have set, I have a pre-made gel for you because the gel run will take around two hours. So here I have a gel for you. You can see there are 5 lanes, and this is my BSA to track the molecular weight. So this is the BSA band, and here I have samples. So you can see for each sample I have different band. The proteins are resolved properly. So this we do to check the QC of the sample so that we know our protein extraction is happened and the proteins are in intact. So you can see now the samples have started moving down. So we have the resolving and stacking gel here. So first the sample will stack in the stacking gel, then it will start resolving. So during the stacking, you have to keep the voltage around 90 volts. Then after moving to the resolving gel, you can increase the volt to 120 volts. In that way, it will resolve. Oh. So you can see the composition of gel here. So usually for the QC check, we are using 12% of SDS gel, but you can increase or decrease the percentage of gel according to your need, because if you have like higher molecular weight, then you have to reduce the percentage, and if you have the lower molecular weight, you have to increase the percentage in that way. So here is the composition of SDS running buffer, and we have the 2x family buffer, that composition as well written here. So now we can move forward with our 2D setup.
All right, good. So, thanks for showing this. And I just hold my way from my office to the lab here. Yes, we can do, as I talked to you, like, the gel can look like for different activities, especially. And you're going to for first dimension separation. And first you're going to place your reference site. Show you some steps here. Okay, so we are first going to take this IPG strip, which is immobilized pH gradient strip, if you can see, very thin. And there are some different pH buffers are already printed, and polites are printed on that. Let's say this one is 4 to 7, which is covering maximum the physiological pH range. You will keep the protein there, and then you will place the IPG strip on top of that. And in this reswelling tray, you will allow the time for the protein to get absorbed in the process known as rehydration. So that will be the first step. And then you can add the mineral oil so that the protein which is already absorbed should not get crystallized with urea and other thing which you add in the buffer. And you leave it overnight. That's a passive rehydration. Alternatively, you can also place that inside this particular isoelectric focusing unit and apply very low volt overnight. That can help you to also do the rehydration step well. So it can be known as active rehydration. So this is the very first thing which you want to do for the 2D gel, to do your first dimension separation, and for that you are going to do the rehydration. Once you've done overnight rehydration, then you will take this strip and you are going to place it in the IEF tray, which will be here inside, and you are going to place some wicks on top on the sides, and then you will place the electrodes so that your isoelectric focusing can be maintained. And this instrument, this setup is also compatible for the dyes, because you can see from outside you are not allowing the light exposure. Quenching is not happening. So therefore, you can utilize the same setup for the fluorescent cyanine dye labeling, and followed by doing the separation with the IEF. So this is the entire setup. Is just showing you the first dimension separation. And once your IEF is done, let's assume that depends on the length of the IPG strip, depends on several other factors. It can take maybe just 10, 12 hours to maybe 20 hours, depending on especially the length of the IPG strip and what voltage setting we have set up to achieve the volt hour gradient. Let's say our IEF is complete overnight, and then you are going to take this IPG strip back, and that you're going to place in the tray again, which will be for the equilibration step. So again, as I talked to you in the theory class or the theory session, that we are going to have the equilibration steps 1 and 2, which we are going to do on this reswelling trays with the DTT, for example, for reduction. And you just simply change the well from one to next one to change the solutions to DTT to the IEA. So this entire thing is making it all prepared for your first dimension separation to happen based on the isoelectric quantity. Once we do that, then I think the next part is again now coming to this type of gel, where we are going to do the second dimension separation. So this is the small electrophoretic setup for the 7 centimeter IPG strip, but this one can be utilized for slightly bigger one like 8, 11 or 18 centimeter IPG strip. We also have some very, very big setup, which is for the 24 centimeter IPG strip. So they can also be utilized for separation. But idea is, if you are to do the separation using 2D gel, you want to separate as many protein possible on the very large gel, which is around 24 centimeter or 18 centimeter, so that you have much better separation of thousands of proteins. And when the bigger, the longer the strip is, you are going to add more amount of protein for separation. So for a 7 centimeter typical strip, if you go 150 microgram, for the 24 centimeter, you can go even 1.5 milligram protein to be loaded. So again, in that way, your overall concentration is much higher and you can get better intensity of the spot for taking it forward. So let's say we have now done the first dimension separation followed by the second dimension, the 2D gel, and then we are going to do the staining of our gel. So if you could see any of the stained gels, I think what currently here we have is one of the SGS gel, which is not a 2D gel, but the process will remain same. We are going to stain with the Kumasi stain and D-stain, or if you want to have more sensitive stain, you can use the Cypher Ruby and other stain which I talked to you. But let's say we have reached till here. Till now, all it looked to us a beautiful abstract image, which showed a lot of beautiful spots on the gel for 2D or some bands on the SGS page. But what we are not able to know that what that protein is, right? So until unless we do addition experiment, we can't tell what each of these bands are, right? So if let's say this is a protein of our interest, which we want to cut, like this one, this band, so then you have to excise that band and then proceed for the in-gel digestion. But by the way, this is the flat comb which you can use for the 2D gel. This is the normal comb for the GSP gel. So again, let's say any of the band which is of our interest, you want to cut and you want to know what protein is. So then you have to proceed for in-gel digestion. So you have to chop out this particular spot or the band from this thing and you keep in a tube and perform this step in the laminar hood so that you are not introducing any contamination, your hair fall or the skin touch, any of those things to be avoided, because keratin, once it is coming on your gel, it is going to be most abundant protein and going to mask every other low abundant protein. So you should be keeping a track of those. Once you do that, then now you're going to take the spot or band in the tube. You're going to first do de-staining step to remove all the stains and then proceed for the same step of reduction alkylation, followed by adding the enzyme trypsin, or you can make a cocktail like trypsin and lysine for the better digestion of the protein. When the protein is digested to make peptides, now that peptide has to be cleaned up, followed by you can do injection in the mass spec. So we are going to take our next session in the next month when we'll talk more about how to take this workflow forward to the mass spectrometry based approach. And that time we will talk to you about the in-gel and solution digestion, some of those hands-on activities, and then followed by how to take those peptides, run in the mass spec and acquire the data. So, but I hope, in this session, you are getting at least the first visual sense of the entire workflow from the sample, moving to the quantification, moving to the qualitative analysis, doing the first dimension separation, doing the second dimension separation, and staining could be done with much more sophisticated way, moving from Kumasi to the Cypher Ruby, or to looking at the even different type of timing dye, fluorescent staining. So all of those are different ways of looking at the gel. I don't know if you have shown the scanner, but you can quickly show the scanner for scanning a gel. So let's say you want to scan a gel. Let's go and look at how to scan the gel.
So here we have the gel scanner. This is a gel scanner. You can just put our gel here and so you have to make sure there is no bubble during the scanning, otherwise your bubble will also come in the gel. Put some distilled water and you have
remove any kind of bubble from the. Now you can just close it, and we have a software called gel scanner. There you can have gel to everyone. If you have any question, you can put in the chat or you can in the Telegram group. We use it like this.
All right, so great. In a very limited time, what we have, I'll just show you once again quickly that what all experiments we have been talking today, that actually does not happen in research so straightforward way, right? So again, this is one of the lab projects which we were doing on the 2D gels. So each step you need a lot of optimization. That's where the difference in doing a lab practical versus the research is. In research, things don't, that you are not optimized. You have to do all the optimization. So in this case, to do the serum proteomics on the 2D gel, we were trying different ways of extraction from the crude serum to triazole extraction, acetone precipitation, TC acetone precipitation. And when we ran the 2D gel, you can see the numbers were moving from 100, 200 to 3 to 500. Then we tried different ways of clean up the sample, like desalting, sonication, and depleting the abundant protein that increased the number to almost 700 proteins. Then we stained the gels from Kumasi stain to silver stain to cyanine dyes, and now those numbers reach to 1600. So think about the same sample, same technology, just trying different ways of extraction, different ways of sonication, depletion, and staining can give you from 300 to 1800 numbers. That can be huge difference. So this is just one of the workflow for doing the serum-based proteomics application using 2D gel or the gel-based proteomics, where a lot of the initial sample processing to be done, abundant proteins to be removed, the lysate to be prepared, protein quantification to be done, followed by we are going to separate on the gels, analyze the pattern of the protein, and then look at their biological relevance. I hope that gives you in a nutshell the comprehensive picture of the research takes a lot of time, effort, and patience. And once you have that shown that perseverance, then I think you can achieve a good protocol, which can help you get the good coverage of the proteome, and then you will have very good ways of comparing the disease versus control sample. I hope you found today's session helpful. If in case you have additional questions and query, please feel free to get in touch with us on the forum or in the Telegram group. We'll be happy to address your queries. Good luck for your exams for the group who is taking the protein and gel-based proteomics course. And we will stay in touch with other groups of proteomics and proteogenomics. And with all of you, we'll look forward to have our next session on mass spectrometry-based proteomics. Thanks to Tia for all the activities and hands-on session today, and wish all of you good luck. And we will be in touch with you over the different forum activities on the Telegram and the your forum for any additional queries and additional information. Thank you very much, everyone.